Okay. Am I still muted?
Nope, you're good, and your screen looks great.
So I'll give you the short version, but yeah, well, thank you so much for having me here and for letting me speak today. So this is actually a project. So it spun out of the Brain Initiative, where a lot of the mouse work was funded, and then we did some extra human work actually in the context of another species comparison hour one that I had, and in context of this NIDA Scorch consortium, which also contributed to funding the human work. And so it's work that was led by a fantastic grad student, Anushka Joglika, who's by now a postdoc, so has moved on, which is why I'm giving this talk. Here are all the authors, and this is hopefully we'll get this someday on a cover here. So basically, what you're seeing here is a depiction of isoforms, which are these black lines here. Okay, here's another one. You see that most of these bulges, which are the exons, are sort of the same. You see here's a special one that doesn't occur here. Likewise, there's a special one here that doesn't occur there. So that's exons that differentiate one isoform from another. And you see that these this isoform is associated to an immune cell type, let's say microglia, and this is more neuronal cell type. So all of this is about cell type specific isoforms, and that's what we're going to talk about today. And this image was done by Luis Salente, who did all the everything that's beautiful in this presentation. Right, so you can think of splicing as basically the process that takes the gibberish that is pre-messenger RNA, locates where the exons are, here in the red boxes, removes the introns in between, and then you get a message, and that's something you can read. Okay, that's the difference between pre-messenger RNA and mature RNA. It was night in October. And so when the last intron was removed, the cell may very well realize that, oops, there's another exon, splice out the intron surrounding it, and now we get a second message. It was night in the lonesome October. Now, these two phrases, they're rather similar. They come from the same gene. Nevertheless, they have a difference in connotation because the second one is out of a poem by Edgar Allan Poe. Okay, so that means here we have a connotation on a message that is not present in the other one. And there's basically, that's one way how you can think about alternative splicing. It's still from the same gene, doing probably something in the same direction, but with different efficiency, or maybe a protein-protein interaction doesn't work anymore. There's other data that I'm not going to go too much into detail, but there are clear examples where one isoform does exactly the opposite of the first isoform. Okay, so these are these night and day differences. They also can arise. And so we basically, when I started my lab, this was our first paper that we put out, where we introduced the idea that you can do long-read sequencing on high-throughput single-cell data sets. And this was led by Ishan Gupta and Paul Collier, but you should look at the author list. There were really a lot of people that contributed to this. And so I'm going to go really quickly over this. So fundamentally, we do long-read sequencing on single-cell libraries. Not single-cell libraries, single-cell cDNAs, it's a slight difference. And therefore, we can then take individual reads, say which cell they come from, and therefore which cluster they come from. So we can say that the top read here is a microglial read, and the bottom one would be a Purkinje cell. So this was done in the cerebellum of a mouse one day after birth. And I should point out that also a year after, or in 2019, colleagues of ours published a similar paper, which is also a great paper to read. And before and after, there were different versions of SmartSeq that also allowed, in the beginning not, but then later, yes, to interrogate splicing with single cell resolution. And especially the Sundberg lab has pioneered a lot of things there. So, based on this first Nature Biotech paper, we also put out this Nature Comms paper with an analysis pipeline and a bioinformatics paper, which basically allows you to visualize these things. And here in every line, you have one molecule. Now, all the molecules that are here in this group together come from one cluster. In orange, we highlighted the alternative exons, and you can see that the neurons basically have a lot of inclusion of all these orange exons. There are 5 of them that are basically 100% included, or 95 to 100. And then there's one that is included to varying degrees. And then when you look at microglia, for example, you see skipping all the time. In OPCs, you see quite a lot of skipping also. So that means on top of the cell type specificity that is conferred by gene expression, we have specificity as well that is, we have added specificity in isoform expression. Now, what's going to be very important today is that, for example, here, the isoform that skips everything, we clearly thought was microglial or OPC, so in summary, non-neuronal. I'll come back to that. So we also then put out another paper, how to do this in frozen tissues, and this work was led by Simon Hardwick, Wen Hu, and Anushka Joglika. But again, contributions by many. I'm not going to go too much into how it actually works today. But that means we can do these plots now also in frozen tissues, meaning in human tissues. And here you see an exon that is very highly included, 100% in excitatory and inhibitory neurons, and not so much, almost nothing actually, in astrocytes, OPCs, and other non-neuronal cell types. And the second one as well. Now, interestingly, one of these exons is really associated to autism spectrum disorder. I forget now which one it is, but that led us really to look into the disease, and we basically now ask how variable an exon is, meaning we have an inclusion value in one cell type and then another cell type, and this is basically the maximal difference between any two cell types. And so high values mean it's very variable, low values mean it's constant, used at the same rate in all the cell types. And so we took from the literature exons that were associated to autism spectrum disorder and those that are not. And we saw that the ones for autism spectrum disorder were very highly variable. The ones for ALS, we also found a significant difference. For schizophrenia, we didn't find it, although I recently saw a paper that also says that you can find it for schizophrenia. I cannot exactly tell you why. I would also like to point out that you should not compare this pair of box plots to this pair of box plots. So it's good to compare this box plot to this one and this one to this one, but not one pair to the other, because these come from very different studies that had different methods. So it's not exactly an apples to apples comparison. And I should also point out that this work on autism, actually, there's work by the Blanco, Geschwin, and Ponting lab, specifically on microexons, that points in the same direction that was actually done before we did it. In ours, it actually came out, this was also there's still a tendency for shorter exons to be very variable, but we see this extending to much larger exons than to microexons. And so you can then look at the inclusion patterns of exons in specific cell types. Now you can ask in disease when it goes wrong, how can that actually be explained? For example, here you have high inclusion in two glial cell types. When it goes down, well, in the disease, well, it must have come from those two cell types. Then we looked also in early development, and this was already part of the Brain Initiative and published in 2021, led by Anushka Joglika, but again, with many authors contributing to it. I don't know why Stephen is specifically underlined, but he also did a lot of things for this paper. And here we looked at early development, 7 days after birth, asking which cell types actually explain differences in brain region-specific splicing. Now, at that point, we also started to use a lot of nanopore data, which has a higher error rate, and generally with long reads, there can be a lot of artifacts, especially when the error rate is high. And so it took us quite a lot of time to make software that we really think doesn't have a lot of false positives. And this project was led by Andrei Przybelsky and Alla Mikenko. And so it's a pure software paper, but that's what we use now. That's what really makes us confident that when we have Oxford Nanopore reads or also PacBio reads, that we don't have false positive isoforms, because previously our impression of the field was that there were actually a lot of false positives popping up. And so with all of this in mind, we basically now started this work for the Brain Initiative, and here are all the authors that contributed to this work. And it's all unpublished, still under review, so please don't scoop us. Right, so what we basically set out to do is to have sort of this isoform consensus over multiple brain regions, and we specifically chose cerebellum, thalamus, striatum, hippocampus, and visual cortex. This was at P56, which was fixed by the Brain Initiative as sort of adult mice. And then we had 3 developmental time points, P14, P21, P28, for the visual cortex and hippocampus. And that means we have now in each sample the variability or the isoform expression for different cell types in different cell subtypes. We also have the isoform variability or expression for the same cell type in development, and for the same cell type in across brain regions. And of course, for the same cell type, that's not always quite the case. You know, we know that some cell types exist only in the striatum and not so much in other brain regions and in the cerebellum and so on. But we can still at a high level compare, for example, excitatory neurons across cell types. And then, you know, with the help of another R1 and which is now, by the way, ending, and this NIDA Scorch consortium, we also did a lot of sequencing in human brains, and therefore we have the dimension of going across species. And so now here's the same gene plotted a little bit differently, and so we have here excitatory neurons at P14, P21, P28, P56, all at the hippocampus, and then just as a comparator, the oligodendrocytes. And you see that there is a lot of exon inclusion in the neurons. However, we already see that here at P28, for example, a lot of the skipping occurs at P28, and we thought these were completely glial isoforms. And yeah, I will come back to this in a little bit. Now, what we now do is we represent the isoform variation in terms of expression across 3 different axes. So here we fix excitatory neurons. Now we can have variation for excitatory neurons between 2 brain regions, between 2 subtypes of excitatory neurons, and between excitatory neurons of different ages. And so here every dot is an isoform, and we simply symbolize the variation pattern of this these isoforms. And so when you're in the center triangle here, sorry, I'm using my cursor here, the center triangle, that means you have more or less equal variation in all these 3 dimensions. Now, equal variation can in principle mean equally low variation or equally high variation. In the vast majority of cases, it's equally low variation here, okay? You see that there's not that much variation by region, a little bit, but much more by subtype, and also quite a bit by age. And we can now play this game. Oh, sorry. Now, the point that is actually really important, that fundamentally we're not so much interested in pure expression variation, because that could simply be caused by gene expression, but we're interested in switches in isoform where one isoform of a gene behaves one way and another isoform behaves another way. And so here in this diagram, what we do is we draw an edge between 2 of these triangles. We want to say, well, for this gene, we have one isoform that doesn't vary, that's what's in the middle, and another isoform that varies a lot by subtype. Now, this edge is very thick because there's a lot of genes like that. And then you have quite a few genes still behaving like this, meaning one isoform doesn't vary, one isoform varies by age, and likewise for subtype and age, and for no variation and region, we see quite a lot, not so much for region and age and region and subtype. And now we can play the same game for multiple cell types. For example, for astrocytes and oligodendrocytes, you can see that it looks actually quite different here. For example, for oligodendrocytes, you get quite a bit of variation between the brain regions, right? So you get very specific types of variation patterns for distinct cell type. So what we're then interested in is to understand the brain region specific variation. And so the first thing that we did was actually to downsample things so that we could actually compare comparisons of one brain region to all the other brain regions with a comparison of 2 replicates. Now, downsampling is a great thing because it allows you to really have a fair comparison. But what it's not so great is really getting everything out of the data that you can, because you're fundamentally throwing away a lot of data. So that means these values on the y-axis are clear underestimations, but we could show, for example, here when you take, I think that's the comparison of astrocytes in the thalamus versus astrocytes in all the other cell types, that it's much more than when you compare 2 replicates here in red. And you see this for astrocytes and excitatory neurons and inhibitory neurons. It's a bit less clear in the other cell types. So now, given that, we here looked at these 3 cell types, and we really took all the data, trying to really squeeze out everything we can out of this data. And you see when on the axis here, you have basically distinct cutoffs for how different it must be. So first we have a p-value that must, of course, be significant and corrected for multiple testing. But then we have an effect size where we basically ask how strong of a brain region specific effect is that. And when we ask for relatively small ones, here we get maybe 40% of the genes. And of course, when you ask for larger effects, it goes down, 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 so that when you ask for dramatic effects, then it's actually not that much. You can see here, though, for astrocytes, if you ask for small effects, you get 40%. Likewise for inhibitory neurons. For excitatory neurons, it goes actually a bit further, and here it's the cerebellum that is the clear outlier, the one that has the highest differences to all the other cell types. Now I'm going to switch gears, and I'm going to stop talking about full-length isoforms, but now I'm talking about individual exons. And we have this concept of extremely variable exons. So it just means that there's one cell type or one condition where the exon is relatively lowly included, and one condition where it's relatively highly included, and we put the cutoff at 0.75, pretty randomly chosen cutoff. But that basically says we have at least one cell type where the inclusion is 75% and another cell type where it's 0% or 25 and 100%. And so for these, we can now have multiple dimensions of variation. We could have this difference could come out of cell types present in an adult brain region. And here we plot now these delta values. Okay, so red means very, very high values. So this would be adult cell type specificity. We could also have cell type specificity early on in development, for example, P14 or P21. We could have cell type or specificity or differences in exon inclusion between the same cell type in two brain regions. That's this here. And between the same cell type at different developmental time points for different ages. And so what you see here is that you have a bunch of exons which actually have very high variability early on in development and in adulthood. Okay, then you have quite a few that have it in adulthood, but did not have it in early development. So they're the ones that gained it, and of course we observe the opposite as well, where some lose it. Now, for the ones that gained it, so that come without cell type specificity, but then gain it in adulthood, interestingly, there's some that only get it in one brain region or a reduced set of brain regions, whereas others get it in all the ones so that there's no brain region specificity. So now, and this was basically all the mouse data. So we did a lot of sequencing in human hippocampi as well. And so what we did here is to plot how much cell type specificity you actually have in human. And here I just want to highlight E3, so that's this large set here, which has cell type specificity in adulthood and in development. And you ask how much it has in human, and you see the peak is really at 0.75, which, or to the right of 0.75, which is pretty much the cutoff we chose in the mouse. So that means in large parts, what you actually observe in the mouse, you can transfer to humans. Of course, not 100% statement, but there's a very strong tendency that this cell type specificity observed in mouse will be present in the human. Now, if you go conversely, you start to define the cell type specificity in human and then ask what's happening in the mouse. And here we're just plotting neuron and glia. You see these exons here that are very cell type specific in human. You look in the mouse and they're not. They're equally cell type specific. But then there's a larger set which still have cell type specificity in human. And you go to the mouse and they don't have it. So that leads me to say that basically there is added cell type specificity in the human brain. And when we just focus here on this set E4, which is the one that gains cell type specificity, but in a brain region specific manner, what Anushka did here was to ask what kind of genes do we actually get. And so what we got was a lot of Go terms related to neurotransmitter levels, neurotransmitter secretion, and synapse. So signal release from the synapse, postsynaptic membrane potential, excitatory postsynaptic potential, and so on. Now, in some sense, the fact that developmentally regulated exons show something related to synapses is maybe not surprising, because of course there is synaptogenesis going on. But that the interesting point here is that affects the genes that do so in a brain region specific way, which means that for those, one really should think about which brain region one is doing. So now I'm just going to come back to this gene where I previously told you that basically in P1 cerebellum in the mouse, we had the skipping of all exons was really a microglia and an OPC isoform, and the inclusion of 5 of these 6 exons was a neuronal one. So now we look here at the same gene in visual cortex at different time points. And what you see is that, P21, everything is all right. It looks exactly the way we like it. All of the inclusion is associated to neurons. And then we look at P28, and all of a sudden, there is huge skipping of huge usage of what we thought was a glial isoform in excitatory neurons. And then it goes away again in adulthood. So that leads me to say that one fundamental point here is, what is a glial or neuronal or whatever finer subtype you want to talk about isoform is really something that also depends on developmental time point. And so now I just want you to remember that here at P21, we had a lot of inclusion of all these exons. Now we go to the hippocampus, the same time course, and all of a sudden, even at P21, we see a lot of this skipping. So it means first the timeline is different, and at P21, what is a neuronal or glial isoform is actually also a question of brain region. Yeah, so now one last point. Here, what Anushka did was to calculate PSI values, so percent usage values for a bunch of exons. And we look here at a number of glial cell types, specifically all the ones on the oligodendrocyte lineage, so OPCs and different types of more mature oligodendrocyte types, and astrocytes. And, when you do this, what you get is a very clear clustering. And the surprise was that the astrocytes and the OPCs all go together. Okay, and then the more mature oligodendrocytes are actually separated. So, that was to me a big surprise. I would have assumed that the OPCs would go with the oligodendrocyte image. So we did the same thing for gene expression, and lo and behold, here it did exactly what I had naively expected, that the oligodendrocyte lineage would basically be separated from the astrocytes. Now, one important disclaimer, when we show these clusterings, that doesn't mean that every single gene or every single exon behaves like that. It just means that here there are more exons that behave alike in the more mature oligodendrocyte types and behave differently in OPCs and astrocytes. But there still may be other ones that contradict this clustering. Right? So it's sort of I don't know if I should call it a majority call or something like that. But if you put these two things together, the way I like to think about it is that, and I'm happy to get comments in that, is that apparently when the astrocyte lineage and the oligodendrocyte lineage split, there seems to be a big shift in gene expression that is associated to this. That's what we see here, and then, but not too much of a splicing shift actually, and then after that, on the oligodendrocyte lineage, after the cells become more mature, so are not really OPCs anymore, then there's a pretty strong isoform shift, which to me was really unexpected. Yeah, with that, I'd like to thank you for listening. A lot of former group members, mentors, collaborators at Weill Cornell, collaborators outside of Weill Cornell. These are the people that do all the magic. The funding sources, and specifically the most part, was funded by the NIH Brain Initiative, and then the human part by NIGMS and our NIDA Scorcher Consortium. These are the authors that are on this paper, so I'd like to thank them. I'm also going to just let this here, the summary slide for a second for this paper. I'm not going to go through it, but if you want to take a picture, you're very welcome to do so. And then I'm going to do the same for all the previous papers that I mentioned that sort of were the basis to do what we did here. Again, you're welcome to take a picture if you like. And there's I'm never quite sure whether I explain things really well. We also recently published 2 reviews, one for Nature Methods and one for transcription, where basically, in case my explanations were really not that good, you can read it and see whether that is better explained than my talk here. And I'm going to leave you with this cover that we did for ENCODE in 2012, which just illustrates the beauty of co-transcriptional splicing. And I guess I should click on the Q&A now, right?
Yeah, so we have
a couple questions for you. Have a look.
Yeah, so the first one is on disease-associated microglia, and it's saying that you see these come up a lot in the context of neurodegenerative diseases. Do you see any patterns emerging related to HIV?
Yeah, we're not there yet. But that's exactly the idea, right? That so in neurodegeneration there are disease-associated microglia, and let's say if one takes the word disease-associated microglia at face value, you would expect them also to be present in other diseases compared to neurodegeneration. We don't know yet, but the hope is that we're going to find something, and maybe also some distinguishing parameters that are not, let's say, common to all diseases, but maybe to HIV. That's definitely one of the major questions to be asked. And the second one, co-occurring isoforms of the same gene within a single cell. We do, but mostly for highly expressed genes. And the thing is that basically, as you probably know, single cell sequencing, in a sense, I always say it's a bit of a misnomer, because we rarely ever reason, for a single cell. And one of the reasons is, well, because we want replicates, so that we want to have some multiple cells that show something, but also because fundamentally we're very limited in the number of UMIs per cell, meaning the 4,000 or 5,000 UMIs that you get per cell are probably nothing compared to the number of mRNAs that are actually present. And that means, likewise, for when we ask for co-occurrence of distinct isoforms, for very highly expressed genes, we do observe this. So there are a couple of examples where that is the case. And there are also a couple of highly expressed genes where you can say, well, there's actually very little co-occurrence of multiple isoforms. But for most genes, for the ones that are not super highly expressed, we can actually make that statement, which is more a limitation of throughput, meaning of UMIs per cell, rather than a biological statement. And the last question, this is it is indeed a Miro cover. At the time was done also by Luisa, who is my wife, and I was a PhD student in Barcelona. It seemed fitting. And the previous one that we showed, this one is actually after Calder, who is a New York artist. So. Yeah. Thank you. I guess I will stop sharing now, right?
Sure.
Yeah. Well, thank you again.
Yeah, thank you, Hagen.
Okay, folks. I'd really like to thank everybody for this incredible set of presentations. Hagen highlighted a bunch of publications, and I'd just like to direct people to our publications website, where if you take a look at some of these pull downs, you can see publications that we've gotten out, and I think I see a total of about 40 that we've generated. So please keep joining us or keep coming to our website and look for other publications that are coming out.